Hi, I'm Palace. I'm HUSO's Director of Service and super excited that you guys can make it out to our speaker series. I have a pleasure of introducing Professor Rockwin. So Professor Rockwin is an assistant professor in the Department of Pharmacology at Northwestern University, Feinberg School of Medicine in Chicago. His lab focuses on both computational protein design and the development of high-throughput experimental to study fundamental protein biophysics, such as folding, stability, dynamics, interactions, and more. He is a member of Northwestern Center of Synthetic Biology, as well as the International Rosetta Commons Consortium for protein modeling and design. So super excited for his really interesting talk. And take it away, Professor Rocklin. I'll give you screen sharing permissions right now too.
All right.
Awesome.
Yeah, well, thank you, Palace, for the introduction and for the invitation, and thanks to everyone who came to hear about protein design. So yeah, like Palace said, I'm a professor at Northwestern. I started the lab. I started at Northwestern about 2 years ago. I'm originally from California. I went to Claremont McKenna College in Southern California, and then UCSF for graduate school. I did what's called postdoctoral research after, so I did a PhD in San Francisco, postdoctoral research in Seattle, and now I'm here in Chicago. And so you know, in science, you learn different things in different places, and kind of bring it all together when you're running your own lab. So I'm going to tell you a little bit. I'm going to give a big overview of protein design and one of the things that's especially exciting in protein design, which is doing high throughput protein design, or designing and testing thousands of proteins in parallel, and what you can learn from that. And I'll talk about this kind of broadly across the field, but also show a little bit of my own contributions and some of the work in our lab. And of course, thanks for everyone for coming, and I'm excited to discuss protein design and research and how to get involved in science at the end of the talk. Okay, so the most important thing that I want to convince everyone of is that right now we're entering a golden age of protein design, and that's because and I'm going to show a bunch of examples of that, but I want to start off just by showing some of the different things that computationally designed proteins can do. So this is an example of a computationally designed protein. It's actually originally an enzyme. It's a naturally evolved protein, but it was computationally modified so that it could break down gluten, which is substance in breads and all kinds of foods that really irritates the stomachs of people with celiac disease. And so this designed enzyme is being developed into a pill that people with celiac could eat to help break down gluten and prevent the damage to their stomach that causes. This is a different example of a computationally designed protein that assembles into this enormous cage. So it's a little bit hard to see because the pictures are the same size, but this thing right here is like 50 times bigger than the enzyme shown here, and it assembles into this huge cage that can actually encapsulate different cargo. And these cages are being developed to deliver encapsulated cargo to different parts of the body. This is another example of a computationally designed protein that was designed to detect the opioid fentanyl. And many of you know that fentanyl is a drug of abuse. And this was again, it was a naturally evolved protein, but it was computationally modified to change its function so that instead of doing what it was evolved to do, it instead binds fentanyl and actually changes its shape in a particular way when fentanyl is present that lets it create a signal inside cells. So these are three examples of using computational protein design to create new functions in proteins. And I selected these from hundreds of examples that you could have chosen about different things that you can design proteins to do. And I put these examples at the very beginning just to get people excited about protein design. But I know that in my understanding, people are coming here with lots of different backgrounds. I was I think they're both middle schoolers and high schoolers here, I think. So I'm not sure how familiar everyone is with proteins. So I wanted to give just a really quick introduction after introducing some of the things that design proteins can do to like, what is a protein? Oh, actually, well, that's going to come in 1 second. Sorry. The first thing, so the reason why we're entering a golden age of protein design is that we have really exciting new tools. And these tools are changing, they're changing how we design proteins, and they're changing the scale that we design proteins at, and they're changing what we learn when we can design proteins. And so in this talk, I'm going to touch on all these three things. And now I'm going to take a little sidestep and talk about what is a protein. So I know this is like review for many of you, but for people that you know, off the top of your head, you're not sure what a protein is. A protein is a long string of what we call amino acids. And proteins have what's called the backbone, which is like that string that runs along the entire protein. And that's what I've colored here in white. And then each amino acid has one, what we call a side chain. And the side chains in this string are colored in green. And there are 20 different side chains that there are 20 different amino acids with 20 different side chains. And you can put those 20 different things along a string in any different order. And these strings can range in size from really short. So what I'm showing you here is a protein that just has 9 amino acids because there's 9 side chains. But these strings can get incredibly long. So really large proteins can have tens of thousands of amino acids, but they all come from those same 20 building blocks. So the different sequences that those amino acids are put together along that chain is what makes one protein different from another. And I know like most of you are probably already familiar with this, but biological systems synthesize proteins using the information, the sequence information that's stored in DNA. So one of the most important functions of DNA, you know, the A's, T's, C's, and G's in DNA, is to specify the sequence that these amino acids go in different proteins. So, you know, DNA plays other roles as well, but one of the most important functions is specifying the order of these amino acids in different proteins. And then the order of those amino acids causes that protein to fold up into a unique shape. So this is the fentanyl sensor that I showed you before, which I think is a string of about 300 different amino acids. Again, there's only 20 different types, but if you can put 300 of them in a particular sequence, and then it will fold up just like this. So protein folding is the process of going from this linear string of amino acids into this folded 3-dimensional shape. And protein design is going the other way around. So in protein design, we have a shape of a protein that we want to make, or a function. And then from that shape that might exist on the computer, we try to find the sequence of amino acids that will fold up into that shape. So protein folding and protein design are kind of two halves of a similar problem. Okay, so I told you just a second ago that we have really exciting new tools in protein design that are changing how we design proteins, and they're changing the scale that we design proteins at, and they're changing what we learn when we design proteins. And now I'm going to touch on each of these three things, starting with changing how we design proteins. So all the proteins that I showed you in that, like these are examples of designed proteins, those are actually cases where we started with natural proteins, where you know, not me or my lab, but different research groups around the world started with natural proteins and modified a small number of the amino acids in them to change their function. And this is basically the way that most protein design has happened for the last like 30 years, is by starting with a natural protein, changing some of the amino acids to enable that protein to do a new thing. And in this fentanyl sensor, I've highlighted, I think maybe 9 amino acids that changed in order to enable this protein to become a fentanyl sensor. Now, in the last maybe especially like 6 or 7 years, there's been an explosion of what we call de novo protein design. And de novo protein design is the ability to design an entire protein fold from scratch. And what I mean by that is, rather than starting from a natural protein structure like this this evolved protein and then modifying a few pieces of it, we actually kind of sketch out an architecture for what a protein structure could be, and then use computer modeling to turn that kind of two-dimensional sketch into a 3D structure that's never existed before and doesn't have to be related to any naturally evolved protein. And then once we have that 3D model, the last step is to then use the computer again to find a protein sequence that we think will fold up into that 3D structure. So the hypothesis here is that if we can create the entire structure of the protein, if we can change the entire architecture of the fold rather than just taking a naturally evolved fold and modifying a few pieces of it, then we can make new proteins that are better customized to the types of functions that we want them to have. And the ability to do this de novo protein design to create entirely new protein structures has really exploded in especially the last 6 or 7 years. So I highlighted changes in how we design proteins. The next huge change is a change in the scale that we can design and test proteins at. So protein design research for 20 years has been really limited by the slow pace of experiments. And I'm going to walk you through kind of how computationally designed proteins were tested 3 or 4 years ago, or this is still a common thing to do, but it's something that is starting to become replaced. So the typical way of testing a designed protein is you have your sequence that you designed on the computer, and then you need to order a gene that encodes that protein sequence. So you need to synthesize a piece of DNA that will specify that protein sequence. So like I said, the information about how to make proteins is encoded in DNA. And cells will have their own DNA, but you can also just chemically synthesize DNA to specify the sequence of any protein that you want. So once you have a sequence that comes off the computer, you email a company that synthesizes DNA and say, please make DNA that has this protein sequence for me. And then you take that DNA and do what's called a transformation, where you basically force it into bacteria, and then you can grow that bacteria in a flask, and all those bacteria will make the protein that you want. So on the right, this is like the flask when we just put some sugar in it before it grows any bacteria. And then this is the flask after a day of growing bacteria. And you can see it's really thick and opaque. And then you bust open all that bacteria and purify out the protein that you wanted to test. And then you do some experiment to see if it folds into the structure that you wanted it to have, or if it has the function that you want to have. And you can see where the slow parts are of this procedure. You have to order a separate piece of DNA for every protein that you want to test. And then you have to grow each one in its own flask. And, it's smelly, and you need shakers to shake these things. And it takes a couple hours to bust open the bacteria and do whatever experiment you want to do. What's really been started to replace this method in the last 3 years is the ability to order tens of thousands of unique sequences all mixed together at relatively low cost. So there's been just incredible improvement in the ability to synthesize DNA and to synthesize large amounts of unique sequences as mixtures. So when you get 25,000 different DNA sequences all mixed together, each individual one is just a few pennies. And then rather than growing each protein in its own flask, you can grow them all together, maybe in one flask. And then you have to use some more complicated experimental method to separate the proteins that were successful from the ones that were not successful. So for example, if you want to study protein stability, then there are different experimental methods that you can use that I'm not going to talk in much detail right now, but we could talk about it in the questions about separating designed proteins that fold into stable structures from designed proteins that don't. Or if you want to study some particular function, you might have a way to separate designed proteins that successfully carry out the function you want them to carry out from designed proteins that don't. And then once you can separate successes and failures, then you can use different experimental methods to determine the identities of the proteins that were in each category. So maybe you started with 25,000 different designs, and then maybe like 2,000 of them folded into the structures that they were supposed to have. And then you can identify what were the 2,000 that folded into stable structures. And then rather than having to do this time-consuming experiment where you purify each individual protein, you get the data that you wanted on which proteins fold and which didn't on this vastly larger scale in a much, much easier experiment. And there are many different ways of performing kind of steps 3 and steps 4 of separating successes and failures and in identifying which were the successes and failures. But a lot of the methods here involve DNA sequencing. So you're finding ways to connect the data that you want to know, like, does this protein fold or not, or does it carry out the function I want it to have, to some kind of DNA sequencing. And many of you know that like there's just been tremendous advances in how easy it is to sequence DNA, and mainly for the purpose of like sequencing people's genomes, but also for scientific research. And if you can connect what you want to learn about proteins, like do they fold or not, to some experiment where that data becomes encoded in sequencing DNA, then you can learn about a huge number of designed proteins in a single experiment. And I'm going to show you a few examples of what this data looks like in a little bit, but there are many different ways of doing this that are not just sequencing DNA, but it's the combination of advances in synthesizing DNA that lets you test tens of thousands of proteins with new types of experiments that let you read out were all these different proteins that I designed successful, that's really accelerating the testing of designed proteins. So let me show you, this is a timeline of kind of different structures that people have designed over the last few years. So, I mean, these are just different shapes of protein structures that don't necessarily have anything special about them, but just different types of shapes that people have tried to design over the years. And one of the things that's always gone along with computationally designed proteins is that a lot of them don't fold the way that you hope they do. So I'm picking out a few examples from 2015 where when each of these proteins was tested the old way, where you buy an individual gene and put it into bacteria and grow a flask for every single protein and then test to see if it folds. And what you can see is that only like a small fraction of the designs actually folded the way that they were supposed to. And that's because the computer models that people were using were the best computer models that we have to design new proteins are really still very approximate. So all these proteins that people test, they look good on the computer, and it's good enough that like some of them actually fold, but the success rate is still not actually that high. And then in 2017, we were able to use some of the methods that I just told you about cheaper DNA synthesis and new ways of testing proteins for folding to really hugely increase the scale that we could test designed proteins at. So rather than testing like 50 or 70 different proteins, we were able to test 15,000 all in very efficient experiments and learn thousands of proteins that folded the way we wanted them to, versus tens of thousands that were still failures. But you can kind of think about just like how powerful that can be in terms of learning the determinants of success and failure. Proteins are complicated. Like all these structures, this kind of circular protein right here has a couple hundred different amino acids, and every one of those positions could be 20 different possibilities. There's 20 types of amino acids, and then you string 300 of them together, and you string 200 of them together, and you could change any one of those positions. So there's a huge number of possible sequences and a huge number of things that could go wrong with any possible computational design. So if you can only test like 20 and then you find out that 2/3 of them don't work, that's not a lot of information to go on to try to improve your designs. But if you can test 15,000 and learn that thousands were successful and more thousands were not successful, that's a much more powerful data set to try to apply statistics or machine learning to understand how do we improve protein design. And I'm going to show you a little bit of an example of that later on. Now, one thing that you might notice about these proteins over here that we could test in a really high throughput way compared to some of these other pictures of protein structures is that these were really small and simple. So many of the proteins on the left are hundreds of amino acids long, but one of the limits of the methods that we were using in 2017 is that we were limited to testing very small proteins. So these are maybe only like 40 or 50 amino acids in a sequence. But the same types of high throughput methods that we were using for very small proteins have actually now expanded in the last few years, both to proteins that have really important functions and to larger, more complex proteins. So on the left here, also in 2017, this is an experiment from the lab that I used to work in Seattle, where we tried designing thousands of proteins, not just to have them fold, but to have them bind to what's called the hemagglutinin protein in influenza. And so this is our small little designed protein in red. And by designing it so that it would stick to the surface of this influenza protein, it actually blocks an influenza infection. And being able to design this protein to stick in just this way is a challenging thing. So we tried designing tens of thousands of examples, and a couple thousand of the proteins were successful. But it was really the ability to design tens of thousands that let us get lots of successes and also learn about what the determinants of success and failure were. Later on, the same high throughput methods that were used here were used to design and test much larger proteins. So now we're testing proteins that are over 100 residues long, over 100 amino acids long, and still testing those on the scale of almost 10,000 proteins at a time. And this last example is a protein that many of you might be familiar with, this really large different colored protein down at the bottom is what's called the spike protein on SARS-CoV-2. And the kind of rainbow colored protein at the top is very similar to this type of design that binds to this flu protein, but instead this is a designed protein that binds to the spike protein on coronavirus and can actually block the virus from infecting from binding to the receptor that it recognizes in the lungs and infecting cells. I mean, the receptors on other cells as well. But basically, this is a designed protein that was discovered to block the binding site on the spike protein on coronavirus, which blocks it from infecting human cells. And so this was actually another really challenging protein to design. This is not work from our lab, but it's from the lab that I used to work in Seattle, David Baker's lab. And this small protein in rainbow was actually discovered in a really large scale experiment where 100,000 different designs were all tested in parallel using the types of high throughput approach that I just described. And they found 105 successful proteins that bind where they're supposed to and block the infection the way they're supposed to. And I think these are currently being tested as therapeutic candidates in animal models right now. So it's been a long way from going from testing, 100 different proteins by purifying each one in a flask and getting a very small number of successes to testing thousands or even 100,000 different proteins and then finding, often large numbers of successes that can be used for machine learning, or in the case of here, at least enough successes that you could start to develop some therapeutic candidates. Okay, so in the last part of the talk, I want to talk about what we can learn when we do these really high throughput experiments and some of the work that's happening in my lab at Northwestern right now. So one of the biggest advantages of these large scale experiments is that we can analyze the data using statistics or machine learning to see what the differences between successful designs and unsuccessful designs are. So I told you that the computer models that we use to make these designs are really, really approximate. And the physics that determines how a protein folds up and whether it will bind to a target, for example, are really challenging to model. And you could use a very complicated, you could try to have, there's a trade-off between the complexity of your model and how fast it is. And you could try to make a very complex, very accurate model, but if you want to design proteins, you need that model to be very fast because the sequence space of proteins is very, very large. So, I've told you there's 20 different amino acids. And if you want to design a 50 residue protein, there's 20 possible choices at amino acid 1, and then 20 possible choices at amino acid 2, and then 20 possible choices at amino acid 3. And if you take 20 to the 50th power, that's a huge number of possible sequences. So you need a very fast model that can search that sequence space. So these models are always going to be very approximate. But with the type of high throughput data that we get from these experiments, we can systematically improve these models. So what I'm showing you here is the very first time we tried doing this experiment. I'm jumping back in time to my work. This is work that I did when I was in Seattle in 2017, where we designed like 15,000 different proteins. The very first time we did this, we got a really small number of successes. So we were trying to design proteins in these 4 different categories. So each of these categories is labeled with these alphas and betas, and that's based on the types of structures in each protein. So the very first time we did this, we tried designing these 4 categories of proteins, and we only got any successes in this one category, this alpha, alpha, alpha category, and the success rate was about 20%. But then by analyzing the differences between the stable and unstable proteins in this alpha, alpha, alpha fold, we could then design completely new proteins that had a much higher success rate. So the success rate in this alpha, alpha, alpha category went up to about 70%. And what we learned from designing this one category was also enabled us to design stable proteins in a different category. So what we learned was actually transferable from one type of protein structure to a different protein structure. And then by doing this 4 times, we were able to make lots and lots of stable proteins in 3 out of these 4 categories. And actually a student in my lab working in the last year has shown that he can now design lots of stable proteins with this 4th type of fold as well. So analyzing the data in this way wouldn't really have been possible if we only had like 5 successful proteins and 10 failures. It was the ability to see hundreds of successes in testing 1,000 at a time or several thousand at a time that let us systematically increase the success in each of these different types of proteins. And that's what's driven the expansion of high throughput. That's what's kind of attracting everyone else in the field to doing protein design in this high throughput way, is the ability to learn and systematically improve by having these really large data sets. So what are we doing in the lab right now? One of the things that we're doing in our lab right now is to try to understand what are we still missing in our computer models. After everything that we learn from these thousands of successful proteins, and like, why do some of our designs still fail? So what you can see on the previous slide is even after we've iterated this like 4 times, trying to analyze each time trying to analyze what's the difference between our successful and our unsuccessful proteins, the success rates of our designs in round 4 in some of these categories is still only around 50%. And I want to emphasize that like all the proteins that we make look really good on the computer, but only half of them actually fold the way that we hoped them, the way that we wanted them to. So like, what's going on? And that's one of the things that we're trying to understand in our lab right now. And we're taking a different approach. And we're still using high throughput experiments, but rather than just designing new proteins and studying successes and failures, we're trying to dig in deeper on individual cases of success and failure. And I'm going to show you a little bit of that data. So one of the ways that you can use high throughput experiments is to just test large numbers of proteins. Another way to use high throughput experiments is to make lots and lots of variants of a single protein. So what I'm showing you here is what's called a mutational scanning experiment. And what these experiments are is the concept of a mutational scanning experiment is to take every single position in a designed protein or a natural protein and to mutate each position one at a time to every other possible amino acid. So this is a protein that has 3 helices. So let me quickly find an example. So something like this protein right here that was designed to bind coronavirus. This is what's called an alpha helix, and you can see that there's 1 helix right there, and then a 2nd helix right here, and then a 3rd helix in the back. So this is a very common type of protein structure, and we design them all the time. And so this is a designed protein that has 3 helices, and on the bottom of this slide, this is all the amino acids in the sequence of this protein from the very first one to the very last one. And so these letters are those 20 different amino acids that make up the sequence of the protein. So the very first position is an R, and the very second position is a K, and the third position is a W. And there's 20 of these there's 20 possibilities of these letters, and they go in this order from the beginning of the protein all the way to the end. And then here on the y-axis, these are those 20 different amino acids. So these are the 20 possibilities that you could have at each position. And then every box is colored according to the experimental result of, if we put that amino acid at that position, does the protein stay stable, or does it become unstable? So white means that the protein stays stable, and blue means that if we put that amino acid at that position, it becomes unstable. So we're only ever changing 1 amino acid at a time. So there's 43 positions in this protein, and there's 20 possible amino acids that we could stick in at each position. You know, if we stick in, like, if we change position 1 so that it's a Q, we keep every other position in the protein the same. And then we change position 1 so that it's an E and keep every other position that's the same. Or we change position 2 so that it's a Q and keep every other position the same. So we're making just 1 change, but since there's 43 positions and there's 20 possible amino acids, there's about 800 different sequences that we could test, where each of those 800 sequences is just 1 amino acid away from what we started with. So what do you see in this data? This is what data looks like for a typical protein. What you see is that at many of these positions, you can basically put any amino acid there and the protein will stay stable. So like at position 1, every amino acid, every box in this first column is white, which means you can put any amino acid at position 1 and the whole protein stays stable. And likewise, you can put any amino acid at position 2 and the whole protein stays stable. But there are other positions that have very specific requirements. So like position 10, which is naturally an L, if you mutate that to a Q or an E or an N or an H, then it becomes really unstable. And that position basically has to be an M, an L, which is what it started with, a V, or an I. So some positions are what we call tolerant, meaning that you can put any amino acid there, and some positions are very constrained. They can only be a specific amino acid in order for that protein to fold. And it's the high throughput experimental methods that let us collect all this data. So like I said, there's 800 different sequences here because there's 43 positions, and we can change each position to 20 different amino acids. So if we wanted to test like these 800 sequences the old way, we'd need to grow 800 flasks of bacteria. But using high throughput experiments, we can measure the stability of all these 800 sequences and see, okay, in this particular design, these are the positions that are tolerant, and these are the positions that are restrictive. And in a restrictive position, like position 28, it's okay for position 28 to be T, S, A, G, or V, but it's not okay for it to be these other things. And these other things have different degrees of badness. Like a D at position 28 is really destabilizing, but an H is only like partially destabilizing. So high throughput experiments let us probe the impact of every amino acid at every position. Now, this is like very standard stuff that actually lots of people have done before. The exciting thing is when we apply this type of high throughput experiment to our failures. So let's take some of the designed proteins that don't fold, and can we change every position and see, well, like why did this protein fail? So this is this is that same type of data, but this is what happens when you take a failed design and try changing each position one position at a time. So the first thing that you see is that now this whole figure is blue. And the whole figure is blue because this is an unstable protein. It's a design that failed to fold. So as you expect, if you take a design that failed to fold and change every position, you still have a failed design. It's still unstable. But what we see is that at a small number of positions, this protein can actually become really stable just by changing like one amino acid at one particular place. So the coloring here is a little bit different. The way this is colored, again, blue is unstable, white means barely stable, and red is very stable. So for example, if you change this position 21 so that it's either a G or an A, G makes it pretty stable, and A actually turns this failed design into a very stable protein just by changing one single amino acid. And likewise, at position 11, if you change position 11, which is normally a V, if you change it to an A, it becomes kind of stable. And if you change it to a G, it becomes actually quite stable. So this is the type of experiment that you would never want to do if you were purifying every single, if you were like growing 800 different flasks and testing each of these proteins to see if they fold into a stable structure. But using high throughput experiments, we could probe every single change to our failed designs and then actually pinpoint like, oh, there was something wrong at this position that caused this design to be unstable. And there, you can see that there's other places here that fix this protein, like besides these two positions. But I've circled these two positions because they're really interesting. If you look at the design structure of this protein, these two positions are actually right next to each other. And what we determined when we got this data was that the way this protein was designed, the amino acid at one position and the amino acid at the other position were actually too large. So they basically bump into each other and prevent this protein from folding into the structure that it was designed to have. But if we change one of these positions from a large amino acid to a small amino acid, then these two positions no longer bump into each other. And now this protein folds into a really stable structure. So you can take this large position and make it small, and then it no longer bumps here. Or you can take this large position and make it small, and then it no longer bumps into this. You only need to change one of these two to remove this clash. And so you might ask, like, well, you have a computer model of how proteins fold. Shouldn't your computer model have known, like, when it designed this structure that those two things were too large? Like, why did it design those two amino acids there if they're so large that they clash with each other? And the answer that I'll tell you is just, it's actually still really hard to make accurate computer models of these things and balance the energy of like, how bad is that clash versus all the other things that contribute to protein stability. And getting this type of high throughput data where we can check every substitution in our failed designs is a really powerful tool for learning how we need to improve our computational models. I want to show you one other really fast example of one of the other things that we're studying in our lab. So proteins don't just have a single structure. They're actually always moving between different structures. And all the different structures that a protein can assume is what we call the conformational ensemble of that protein. And everything that I've shown you so far has basically been about designing proteins to fold into a single structure and using high throughput methods to test whether a design folds into that structure at all. But one of the kind of holy grails in protein biophysics has been to be able to characterize all the different structures that proteins form, and to be able to do that in a high throughput way. And this is one of the things that our lab is working on right now. And we're using a technique that's called hydrogen deuterium exchange. And so what this technique is if you take a protein that's in water, and you move that protein into heavy water. So heavy water is water where instead of hydrogens, you have deuteriums. So deuterium is, just an isotope of hydrogen that has an extra neutron. So this is D2O. It acts just like H2O, except it's heavier. If you put a protein that's in water into D2O, then there's a particular hydrogen on the protein that's going to come off. It's going to come off that protein and it's going to be replaced by a deuterium. And every amino acid in the protein has one, it's called the backbone amide hydrogen, and it will come off and it'll be replaced by a deuterium from the solvent. And in order for this to happen, that position on the protein has to be on the surface. It has to be on the surface of that protein where it touches all the water molecules that are around. So I've put a bunch of Ds on this protein kind of on the outside of it. But the positions in the protein that are more on the inside, or that are in what we call the secondary structures, in the alpha helices and the beta sheets, those positions actually don't exchange for deuteriums. And in order for them to exchange, that protein has to go through what's called a conformational change. It has to change its structure so that a piece of it that was previously in this helical structure is now kind of open and exposed to solvent, exposed to all the D2Os that are surrounding the protein. And only then will the H's come off and be replaced by Ds. So this is a very old technique for studying proteins, and it tells you which parts of a protein are kind of on the inside and which are on the outside, and it also tells you what are the energetics of, like, how often do all the different pieces of the protein open up and expose themselves to the water, the environment. So different parts of the protein have, the whole protein has some stability that determines whether it's folded or unfolded, but individual parts of the protein have local stabilities, and this technique lets you observe the local stabilities of all the different parts of the protein. And what we're working on in the lab right now is a high throughput way, basically, of measuring this for thousands of proteins at a time, so that we can determine, just like we used our old data on stability to determine how do we design more stable proteins, we can use this type of high throughput data to learn what are the causes of these types of conformational fluctuations in proteins. Okay, so that's the last bit about work in our lab. And the last thing that I'm going to tell you about is a few different ways to get involved in research. So one of the first and most straightforward way is with scientific games that actually really contribute to scientific research. And one of those that I'm most excited about is a game called Fold It, that lets you actually design completely new protein structures using a game-like interface. And Fold It, actually, there's a huge number of different things to do in Fold It besides designing proteins. Fold It is actually used to optimize protein structures and to design new proteins. And so within the Rosetta community, which is a community of protein designers that I'm a part of, there are research projects that basically go back and forth between PhD students and postdocs in research labs and everyone around the world who plays this game. And one of these was actually just published in summer. Well, okay, so this is summer of 2019, where they took about 100 different proteins that were designed by Fold It players and tested all of them for folding stability in the lab, and learned which ones fold and which ones don't, and use that to kind of improve the tools in Fold It to help people design more stable proteins. This is just one example. There's actually like 10 different studies that have been published that happened through collaboration between protein design scientists and Fold It players. And so I really encourage people that are interested in this work to try out Fold It. Eterna is another type of game in the same class, where instead of designing protein structures, players design RNA structures. And this has actually gotten really important recently because designing RNA structures is really relevant toward designing COVID vaccines. And so one of the Eterna projects is basically to design COVID vaccines with improved stability. And they're really taking advantage of the community of Eterna players to make that efficient. Okay, so besides those types of games, how else do you get involved in research? Look up professors in the area where you live and email them. Professors very commonly will have paid or volunteer positions for high school or college students. And what I want to encourage you here is to be persistent. You might need to email like 5 or 10 or 20 different people before you find someone who can say like, sure, we have a paid position in our lab or a volunteer position in our lab. But if you're persistent, I think you can find a lab to work in. Once you're in college, it's almost always possible, it's usually possible to get work study money to do research work or to find programs that pay for undergraduate research. And many, not all necessarily, labs will also accept volunteers. And again, the important thing is to be persistent. And you might have to email 20 different professors before you find a lab that says, sure, we have someone that can mentor you. But I think you'll find someone if you do this. And then another type of program to be aware of is what are called REUs. So these are what are called research experiences for undergraduates. And there's a huge number of REU programs all around the country. We have one here at Northwestern that's focused on synthetic biology. And there's also a Rosetta Commons REU that's really focused on computational protein design. And so typically for these REUs, you would apply in December or January, and then these support full-time paid research in the summer. I want to just give 2 last pieces of advice for anyone looking to do research in the future, or just, I think this is good advice in general. Do your best to find mentors who are, these could be, who are 2 to 3 years ahead of you along the type of career path that you want to have. So if you're a high school student, if you're like early in high school, this could be a high school senior. If you're a high school senior, this could be a freshman or sophomore in college. If you're an early college student, this could be a college senior. It's really valuable to have people that are just a few years ahead of you along the type of career trajectory that you want to have, that you can ask like, well, what are the types of opportunities that you're seeking out? How did you find this? What are you doing right now? And I've tried to do this basically. Here I am, I'm like 36, I'm a professor at Northwestern, but I still learn from people that are like 2 to 3 years ahead of me. Like, how do I reach the next step in my career by learning how you've walked through this, by learning how you navigated the point in your career when you were my age. So like when I was a college student, I try to learn from first year graduate students. And when you're early in grad school, you try to learn from the senior graduate students. So you'll meet these mentors in all kinds of different settings. But do your best to acquire people that you can learn from. And the other thing is that there's a huge number of different paths to success, and that people become scientists in lots of different ways. So just for myself, like when I was in high school and middle school at the stage that you're at, I didn't do anything connected to science. Like when I was in high school, I played in the band and I went to speech and debate competitions. So I didn't like do research in labs or like play folded games or whatever. But in college, I became interested in research and started doing research, and then went to grad school, and here I am. And there are people that I worked with, like a lot of the best scientists that I've met in when I was a grad student or postdoc are people that didn't start grad school until they were 5 years out after graduating college. Like one of my colleagues in my lab when I was a postdoc went into college as a like music composition major and was a serious like musician for a really long time, and then only started doing his PhD in science a lot later, and now is quite successful. So there's not just one track where you decide to major in science right away in college, and then you start PhD right after college, and then you do a postdoc or whatever. There's lots of different paths. Okay, so sorry for speaking a little bit longer than planned, but I'm happy to take questions about anything that I talked about. And thanks again for the invitation.
There are a couple of questions in the chat,
well,
if you want.
I'll take your questions. I just want to thank people who contributed to this work. Taeyun is a grad student in my lab who contributed some of the design proteins. Hugh Haddix is a postdoc in David Baker's lab who did some work, who's working with me on understanding determinants of like rescuing the unstable proteins. I think those are the most important things to highlight. Okay, cool. Questions in the chat.
Would you prefer for me to read them directly, or do you want to read them?
I got it. And then I'm happy to have people unmute their mic if they want to just ask questions that way. Was Hong 2016 a Tim barrel? Yes, it was. Great observation, Annabelle. How does the kind of machine learning your lab rely on differ from the AI-based algorithms like AlphaFold 2 that are being used to predict protein folding? Different tasks. So different types of machine learning methods are useful for data at different scales. So one of the biggest advantages of AlphaFold, so for people who aren't familiar, AlphaFold is an incredible machine learning model for predicting protein structures. And the training data for AlphaFold comes from like over 100,000 different structures of proteins that have been solved over the last like 50 years. And when you have hundreds of thousands of data points, or when you have like 200,000 different examples, you can use really sophisticated machine learning methods that can then make really powerful predictions. The types of data sets that we're working with are often on the scale of like maybe tens of thousands of examples, and we're also trying to learn different things. It's a complicated question, but to just highlight one other thing. Different machine learning methods, it's not just like when you have a large amount of data, you do this, but when you have a small amount of data, you do that. Different data sets differ in terms of, for example, how diverse the examples are. So for example, if you have like 100,000 examples that are very different from each other, then that's a much more useful data set to learn from than if you have like 1 million examples that are all very similar to each other. I don't know if this question would take a very long answer, but maybe because the progress made in structure prediction by AlphaFold is really, really exciting, I think I'll just highlight one example of a difference between AlphaFold and the type of work that we do, and why there's still like a huge amount more research to do. One of the basic assumptions of AlphaFold is that the sequence that it's given is going to fold up into a stable structure. And that's because the whole nature of the competition that they compete in is giving protein structures that have already been solved and seeing if AlphaFold can determine what those structures are. When you're working with designing proteins, a lot of what I showed you was that many of our designs that look really good on the computer, they actually don't fold into any stable structure at all. And if you gave those sequences to AlphaFold, I'm reasonably sure that AlphaFold would predict them to have the same structures that they were designed to have. Because AlphaFold, for example, isn't trained on like proteins that don't fold up at all, because largely like that data doesn't really exist except in terms of what's coming out of the types of high throughput experiments that we're doing. So if you want to learn like why do designs fail when you don't already have this starting knowledge that a protein is going to fold into a particular structure, AlphaFold hasn't solved that problem basically. So that's a little take on why there's still a lot more research to do besides even with the success of AlphaFold. But it's really an incredible achievement to be able to predict the structures of protein of these of lots of different proteins once you know that those proteins have a structure.
I think Annabelle has her hand up. You can go ahead.
Okay, so we'll take Annabelle's question first, and then there's another question in the chat.
I just had a question about like the logistics of the hydrogen deuterium like experiments. So once like the deuterium like replaces the hydrogen, how does one like detect where it is like on the protein?
Yeah, that's a great question. So the type of experiment that we do is what's called mass spectrometry, which is basically a way of measuring the masses of, you could measure the mass of one protein, or the way we do it, we measure the mass of thousands of different proteins that are all in the same sample. And, you know, because the deuterium has a different mass than the hydrogen, you can see for every different protein in the sample how it picks up, the speed that it picks up additional mass, which tells you about the stability of that protein. And then if you break the protein up into little pieces, you can see, oh, that extra mass came on here in this part of the protein. And that tells you about the local stability of each, you know, each small part of your protein. That actually measuring the changes in mass is only one way to observe hydrogen deuterium exchange, but it's the method that's most amenable to a high throughput approach where we can look at thousands of proteins at the same time. And then Dan Nee asked, can you give some examples of things you learned from the alpha-alpha-alpha fold that transferred to and improved the success rate of other structures? Yeah, that's a great question. So the types of things that we learn from our experiments are not necessarily what are the determinants of protein stability. In a large part, we know most of the things that determine protein stability. What we learn from our experiments are what are the areas where our computer models are most wrong. So our computer model is trying to balance how many different things contribute to stability. So some of the things that contributes to stability are the hydrophobic effect. So the fact that certain amino acids want to stay away from water, electrostatics, hydrogen bonding. These are lots of different things that contribute to protein stability. And we have a computer model that tries to balance all of them. And then one of the first things that we saw with that alpha-alpha-alpha fold was that the stable proteins generally were the ones that were more hydrophobic and kind of less relied on polar or electrostatic interactions for their folding stability. And that's not because those interactions aren't important. It's just because when we designed those proteins, our computer model was maybe a little bit too tilted towards saying like these interactions are really strong and these interactions are weak. And so by seeing that we needed to basically have proteins that relied more on the hydrophobic effect instead of other interactions, we could sort of tilt the computer model back in the other way. And then that appeared to be transferable because when we tried designing other types of proteins with that changed model, we were more successful. And again, all those different things are important for stability. It's just how is our computer model most kind of screwing up the balance? David, are you trying to ask a question or?
Yeah, hello.
Hi.
Hi, Dr. Rocklin. Yeah, thank you so much for giving the talk. De novo protein design has so many like incredible, like imaginable as well as like unimaginable applications in the world. I guess my question would be more on sort of like the practicality side. Sort of a lot of the testing that you talked about seems to be more on like focusing on stability and specificity. Are there also like high throughput tests that could verify like off targeting, or rather like the lack of off targeting with these proteins and potential applications?
So if I understand what you're saying, like if you design proteins, you want to know not just like, does it bind to the target that you're specifying, but also does it interact with other things in the body that you might not want it to interact with?
Exactly. Yeah.
there the answer is yes, but it's not always perfect. So there are definitely high throughput approaches where if you have like 100,000 different designs, you can profile all of them using a high throughput experiment to see which ones are most likely to be what we call promiscuous, meaning interact with lots of different things and not just the thing that you want. It's not perfect. But then, there are high throughput ways to look at general promiscuity. And then if you had a specific off target that you really wanted to avoid, like we need to inhibit protein A, but we don't want to inhibit protein B, then you can screen both of those in high throughput pretty effectively.
Thank you.
Yeah.
I have a question.
Yeah.
So how do you, I guess, like, what would you suggest in terms of like developing computational skills in order to do like the work that you do?
Yeah, that's a great question. So I actually never took a computer science class. And I'm not like a programming savant either, where I just like became a genius self-taught. I'm like a very low average programmer, but it all came from just like fiddling around with stuff myself. But I do really recommend programming and scripting courses, or computer science courses, data science courses. There's actually a huge amount of resources online about how to learn like ways to teach yourself data science. Math is really valuable. One of my biggest regrets from college is that I only took math through. So, like a normal math sequence is like 2 semesters of calculus and then multivariable calculus and then linear algebra and other courses. I never took linear algebra. And then when I got to graduate school, I realized that it was actually incredibly useful for basically everything, and had to kind of teach small parts of it to myself. So I'd totally recommend taking math through linear algebra. I think most colleges are going to have computer science courses targeted at biology and bioengineering majors that are probably really good, and would probably be even better than computer science courses targeted at computer science majors. But I guess one last piece of advice that I'll give is that when I was a second year in graduate school, I got really good advice from a visiting professor who said that in the end, the specialists end up working for the generalists. And I try to keep that in mind, which is that like when you do a PhD, you like the whole point of PhD research is to like really specialize in one particular thing, but you have to find ways to push yourself to like for example, having extreme technical skill is really, really valuable. But I want to encourage people like not to only, like if you're interested in bio, don't only focus on being like a machine learning, like computer science major. Like it's still really great to take the broad scope of biology classes and other things, and to try to stay like broadly interested in lots of things rather than specializing too narrowly, because I think it's being able to be kind of a generalist that lets you connect lots of different things and do science that I find, I guess, the most exciting. Okay, cool. Well, anyway, it was great to meet all of you. I'm happy to like, follow up with anyone who wants to email me about any particular subject. I have to go pick up my daughter from daycare, but thanks everyone for coming, and great meeting all of you.